So I will be talking about the genetics and testing approaches for facioscapulohumeral muscular dystrophy, which you all obviously are aware of in, either there's an affected family member or you know somebody or you're involved in some way. So for the remainder of the talk, I will be saying FSHD. So, but let's just go take a step back and just look at what are we talking about here. So, as June mentioned, we use the term genetics. And so essentially, let me just kind of like explain what is genetics. So genetics is a discipline of biology where we are looking at heredity and variation in living organisms. And how do we go about assessing it, essentially? So this deals with the molecular structure and function of genes. And when I go a little bit later into my talk and I talk about the loci and the genes of interest, then you'll have a little bit better understanding. And also, what is the pattern of inheritance from parent to offspring? And that's where when we have any genetic disorder, it becomes really important to understand what is the mode of inheritance and what is the variation that we see. All of this cumulatively essentially becomes part of genetics. But genetics is also used to explore genetically or inherited diseases. And this is what we are trying to understand here, the complexity of FSHD, how it manifests, what are the causative, reasons. And so we can talk about it in a little bit more detail. So as I mentioned, we talked about genetics and then we come to DNA. So DNA, which most of you might be familiar with, stands for deoxyribonucleic acid. And it is actually the hereditary material that makes up the genes. It is made up of 4 letter codes called bases: adenine, thymine, guanine, and cytosine. And you can see that how they pair. So you can see that adenine pairs with thymine and guanine pairs with cytosine, which you can see in these color coded. And this is how we have 2 strands of DNA that actually are complementary to each other because of this phenomenon. And this is what our hereditary material essentially is. So as I was mentioning on the first slide, I mentioned the term genes. So what actually are genes? So genes are discrete packages of genetic instruction for growth, development, and maintenance. So when you look at, okay, this is our chromosome, which you will hear when I talk about FSHD and the chromosome that we are testing. And chromosome is actually packed DNA along with protein, in just a very simple form. And the DNA is made up of numerous number of genes. So approximately 25,000 genes in the human genome, and this is only 1% of the total genome. So the genes only comprise of approximately 1% of the total genome. And then the the DNA itself, as I just mentioned, is made up of these bases, these 4 bases. And these genes are also made up of exons. So when you hear the term exons, these are the coding regions of the genes, and introns, these are the non-coding regions of the genes. So just very simple, basic terminology for DNA, genetics, and genes. But when I mentioned the mode of inheritance for any genetic disorder, so what that essentially translates into is how are traits passed on to the next generation. So if we look at the broad buckets, there are 3 most common types of inheritance. One is called autosomal dominant, the other is autosomal recessive, and then we have X-linked. There are some other forms also, which we will not go into details, but for the purpose of today's talk, because we are mostly focused on autosomal dominant inheritance for FSHD, which I will explain in a little bit. So that is what we want to actually understand. What does autosomal dominant mode of inheritance mean? So FSHD, there are some certain facts. Number 1 being it's the 3rd most common form of muscular dystrophy. The incidence worldwide is 1 in 10,000 to 1 in 25,000 individuals. And there are 2 types. One is called FSHD1 and the other is called FSHD2. Now, FSHD1 is pretty common, which we see essentially in 95% of patients, but 5% of patients could be having FSHD form 2. The age of onset can vary, but typically you will see clinical features manifesting around adolescence. One of the classic features of FSHD is this scapular winging that you see in this image of this patient, and some of you might be aware of it. As I mentioned, depending upon how you are associated with the FSHD society. But the main symptoms of FSHD are scapular winging. There can be some facial weakness, some atrophy of the upper arms, abdominal weakness, lower leg weakness, chronic pain, retinopathy, and also some sort of hearing loss, high tone sensorineural hearing loss. So going back to that, the disorder itself comes under autosomal dominant inheritance. Let me just explain to you with the help of this cartoon what this means. So autosomal dominant means that a single gene variant leads to condition or risk for the condition. So what does that mean? If one of the parents is affected, there is a 50% chance of each offspring to inherit the disorder. So if we look at the cartoon on the right-hand side, the light shaded gene, which we are saying that little box is a recessive gene on the light shaded chromosome, and on the dark chromosome is the dominant gene. So you can see that the mother has both light forms, so she's unaffected. The father has the dominant gene. So now if we look at how the segregation in the next generation is going to take place, you can have 2 affected children and 2 unaffected children just by mode of these permutation combination of how these chromosomes with the dominant and recessive genes are going to pass to the next generation. So FSHD1 has an autosomal dominant inheritance pattern. This has been well established, right? So there's only one variant that can lead to the condition or the disorder or the clinical manifestations. Now, the genetics of FSHD is extremely unique. Okay, so this is where I think some of you may know this, but let's just go into this. This is from one of the world's experts. They have been working on FSHD and on this locus for a very long time. This is a paper that was published in 2007 by Lemers et al. And what you see here is that we have 22 sets of autosomes, and we have X and Y chromosomes. So humans have 46 chromosomes. On chromosome 4, right, so from 1 to 22, on chromosome 4, at the band position Q35, there is this repeat cluster D4Z4. And it is a 3.3 kb unit repeat sequence that you can see, which is called a repeat array, which is present here. In a normal situation, in a normal individual, there are 11 to 100 units of these repeats on chromosome 4, right? If you have that, if you have the normal range, then you will not have FSHD. However, there's a little bit of complexity here. If you see towards the end of the cluster, you see this little rectangle that says A/B. This is a polymorphic region on chromosome 4, which either will be A or B. So now it becomes really interesting to understand that at what time will the disease actually manifest. So this happens because this array cluster actually contracts. And if you have between 1 to 10 units, so now you see, so from 11 to 100, the reduction goes into 1 to 10. And in the presence of the A polymorphism is when FSHD will actually manifest. Now, if you do have the repeat contraction, however, it is with the B polymorphism, then it will not manifest into FSHD. And this is because of how this repeat cluster in our genome, creates an environment for this toxic protein to be formed, and the DUX4 protein, and leading to the disorder. Now, the other interesting thing is that on chromosome 10q, there is exactly the same repeat cluster. So traditional technologies really struggle with this kind of differentiation. Can you accurately differentiate between chromosomes 4 and 10, number one? And number two, when you are able to do that, can you accurately haplotype and determine if this is the A or the B polymorphism for you to have an accurate genetic diagnosis of FSHD? So this is, I'm trying to explain in a very simple way, but there's a lot of complexity involved over here. There's methylation patterning and a lot of other SNPs and a lot of other things that are there in literature. But for simplistic understanding, what we are trying to understand here is determine the D4Z4 array contraction and also the presence of the A or the B polymorphism in patients that have FSHD phenotype. So traditional FSHD testing, as I was talking about in a little bit, it's very challenging, as I've just explained to you how the unique phenomenon is. And there are numerous labs that have offered traditional FSHD testing by cumbersome technologies, and there are challenges to accurate diagnosis, and also in terms of a fast turnaround time. One of the methods is called Southern blotting. And what it tries to do is, again, determines the repeat contraction along with haplotyping of the A or the B allele. So you can see on the left side of this flow chart, if you do have the repeat contraction, then you will have FSHD. However, if you have the contraction, as I just mentioned in the previous slide, but you do not have the A4QA haplotype, then there will not be any FSHD manifestation. Now, the complication becomes when you come on the right-hand side, that if you have a normal range of your repeat array, however, if you have one of those in the 4QA polymorphism, then methylation assay can also ascertain if there is FSHD type 2. Now, the methylation assay can only ascertain if there is another gene, SMCHD1, and I'll talk about it in a bit, which is on chromosome 18, that variations in SMCHD1 along with hypomethylation and the presence of the permissive allele, that means one of the 4QA alleles, can lead to FSHD2. So which is the rightmost flow chart of this diagnostic workflow that you see. Any other combination that you see in the middle, whether the hypomethylation is not there, the sequencing variant is not there, the 4QA allele is not there, will not lead to FSHD1 or FSHD2 phenotype. So as I mentioned, the other thing is that when I spoke about autosomal dominant inheritance, when you now think about not only do you have the 4Q D4Z4 cluster to think about and deal about, but also you have this gene SMCHD1. So this actually converts this into digenic inheritance, because now you've got two genes that are playing a very crucial role in FSHD2 phenotype manifestation. So there are a few publications on the slide where you can see that it is very important to discern between chromosomes 4 and 10 when you're trying to look at or detect these D4Z4 arrays, and also how this contraction leads to an abnormal expression of the DUX4 protein that actually then causes the facioscapulohumeral muscular dystrophy phenotype. Then you can also see in the top right-hand side screenshot that the digenic inheritance in presence of the D4Z4 permissive allele, or with the A polymorphism, causes FSHD2. So a lot of research has gone in, a lot of studies have been performed. However, at the bottom right, you can see that there is this new technology or optical mapping or whole genome optical mapping that also allows for accurate quantitative measurement of these repeats in an effort to have an accurate and a faster diagnosis of FSHD. So keeping that in mind, at PerkinElmer Genomics, we adopted this a new technology, whole genome optical mapping by Bionano Genomics. And they have this very unique instrument, which is called the Sapphire instrument. So let me just explain to you what this new technology does. So first of all, if we think about sequencing, we've got very short molecules. Those short molecules, when we try to align them back to the reference genome, we have issues. What kind of issues do we have? Will it go back to chromosome 4 or chromosome 10? We don't know, because these are really short sequencing reads. So this is one of the limitations why sequencing technologies are not useful for a disorder such as FSHD. Now you think about, well, let's assume that we have a technology that can give us long molecules. Now these long molecules can tell us, okay, is this chromosome 1 or 4 or chromosome 10? So this is what we're trying to do over here. So if we start from the top left, this technology utilizes a very special DNA isolation method. So as if you remember, we have to look for these D4Z4 array repeats on our chromosomes or on our DNA. So these high molecular weight long molecules are isolated and they are labeled with specific sequences across the genome. These labels are very unique and very specific, and they have a very unique pattern for every chromosome. And this has already been well established. Now, this labeled DNA is then transferred onto a little chip, and the chip has nanochannels. And those nanochannels allow the linearization of these long molecules. And because these fluorescent labels are on there, then in the Sapphire instrument, images of these labels are taken. And then these images are then converted into molecules, unique molecules or digital molecules, which are then interpreted as which part of the genome are we interrogating. And then these long molecules or maps from these images are used to look for structural variation. And in this case, we are talking about either a normal repeat range or we are talking about a repeat contraction, which is what is of interest. So this is a very new technology which allows high throughput, high resolution imaging of very large DNA molecules. And this is what we really need to know. And this is how we can differentiate between chromosome 10 molecules, chromosome 4 molecules, and chromosome 18 molecules. So how does this look like? So what you see in the top panel, actually let's look at the bottom panel first. So what you see is this light blue region, which is your chromosome 4 DNA. And then the purple block is the D4Z4 repeat. And then you can see the next part where you see this red box on top of the blue box is whether it's the A polymorphism or the B polymorphism, right? So the bottom image is actually showing us a control sample without FSHD, where you can see that this is a normal repeat range between 11 to 100. And this has been well established by running normal individuals and normal cell lines and then Coriell cell lines. So we actually know based on the size and these labels that what plus minus 1, what the repeat range would be. What you see at the bottom, those little yellow molecules, those are actually individual DNA molecules from a patient sample, which have been labeled. So you can see the length of those molecules. So you can see a little variation. See if I can use a pointer. Okay, so what you see here is that these are long molecules. And these are the labels on those molecules. So you can see that this molecule over here starts from this label and extends all the way up to the end. So this is a huge advantage of using this technology. And then by aligning all of these molecules together, we create the map of the patients from the patient sample. Now let's look at the top panel. So this is a sample with known FSHD phenotype, and what you can see over here, that if they are aligned equally, we see a repeat contraction. And because of the size of these repeat contractions, plus minus 1, we can accurately type what is the number of those repeats. And again, the labels for the A and the B polymorphisms are unique, so we can accurately type that whether this is the A allele or the B allele for an accurate diagnosis. So in our study, what we did was we collaborated with one of our clinicians in India, Dr. Satish Khadilkar. He's very well known in India, and all of these are his patients with FSHD, or suspicion of FSHD. So you can see that there were 14 patients that we included in this internal study. And what you see highlighted in patient number 2 and patient number 10, that one of the key features that are missing over here is actually scapular winging. But they had some muscle weakness and some limb girdle phenotype, so they still had some muscular dystrophy. And for the most part, we included all of these. And you can see this family also of 1026, where we had 4 affected individuals from the same family. So we were able to get these clinical samples and then perform the internal study on this whole genome optical mapping technology. And we were able to accurately give a genetic diagnosis. So our summary and results from our internal study is that, first of all our clinical samples, 14 out of 14 clinical samples demonstrated 100% concordance. And we performed this study at 2 sites, one in the PerkinElmer Genomics lab in Pittsburgh and one in San Diego. Also, 12 out of the 14 clinically diagnosed FSHD patients demonstrated the contracted allele between the ranges of 2 to 8. Remember, I said it has to be between 1 to 10. Now, the other 2 patients, they could have an overlapping phenotype. We are pursuing with methylation studies and also sequencing to see whether they could be in the FSHD2 phenotype, or maybe they have overlapping features of just muscular dystrophy. In our internal study, we also saw 100% analytical accuracy and precision, and our performance metrics were excellent. So our diagnostic workflow at PerkinElmer Genomics is that we will perform whole genome optical mapping if a patient comes in with a clinical suspicion of FSHD, and we will perform this whole genome optical mapping and for QAB haplotyping and the ability to separate them from chromosome 10. And if we see the repeat contraction, then we will give a diagnosis of FSHD1, a likely diagnosis if it is consistent with the phenotype. However, as I've mentioned initially also, 5% of patients may have FSHD2. And if we see that there is the 4qa allele, but not the contraction, then we can recommend SMCHD1 sequencing to find sequencing variants. In fact, we've seen 2 cases with large deletions of chromosome 18p. And at that time, we would recommend that methylation studies be performed. We are not doing those currently. There is some methodology in development right now, but they are likely FSHD2 phenotype. So this is what our PerkinElmer Genomics testing workflow is. Also, as I was telling you, there is a lot of phenotypic overlap when you talk about neuromuscular disorders and a lot of genes that are reported in literature. So at PerkinElmer, we have a comprehensive neuromuscular disorder panel. And because FSHD2 might overlap with some of those, SMCHD1 gene testing is actually part of our neuromuscular disorder panel. And this might be a better testing approach. However, if there is some overlapping phenotype and there is a suspicion, then testing can be performed or they can be combined, and we can look for both the sequencing variant and the repeat contraction using sequencing and the whole genome optical mapping technology. So now with this complexity and understanding and looking at what our data was on our internal study, we are clinically testing it. We are getting samples from around the globe. We are a global lab. So who should consider getting tested? So if you are suspected to have a muscular dystrophy, or your child, or if you have had previous genetic testing that had certain limitations that was unable to provide a diagnosis, or if you or your child current diagnosis does not fully explain the symptoms, or you have a family member that has been diagnosed with FSHD, then all of those individuals can consider getting tested. And you can see again over here the snapshot of the technology that we are using in-house, that we have developed and validated as a lab developed test, as a clinical test, and we will issue clinical reports. So how should patients go about, or family members go about getting tested? So the most important thing is that the test has to be ordered by a medical provider, so, such as a physician, nurse practitioner, physician assistant, or a genetic counselor, and consulting with specialists in genetics. So genetic counseling is extremely important, or consulting with a geneticist is extremely important for you to decide if genetic testing is right for you or your family member. So how much time does it take, and what is the right sample type? So this is a blood test. We want peripheral blood, 3 to 5 ml of blood. And the turnaround time is 8 to 12 weeks right now. And this could be because they could be tiered testing, but we attempt to release these reports as soon as the data is available. But this is what we have right now. With that, I would like to acknowledge my entire team and the collaborators. So as I mentioned, Madhuri Hegde, who is the Vice President and Chief Scientific Officer of PerkinElmer Genomics, Dr. Suresh Shenoy, who played a very important role in the internal validation study, Dr. Rajiv Rose, who's in India. He's one of our technical directors over there, who played a very important role in sample aliquoting and collection for these samples from India. The 3 clinicians, as I mentioned, some of you might know Dr. Khadilkar, Dr. Dastur, and Dr. Gaitonde, who actually were able to get these patients in, get the consent from them for us to do the study. And then the entire team from Bionano Genomics that you see that really helped us in doing the inter and intra-site validation and test the performance metrics for us to validate this technology. With that, I will take any questions, and and hopefully this was informative and educative for everyone. Thank you very much.
Thank you so much, Dr. Chaubey. This is June again. So people in the audience, if you have questions for Dr. Chaubey, please post them in the Q&A link down at the bottom of your screen. So I had a quick question just for clarification. You mentioned towards the end of your talk, tiered testing. Can you define what that means?
So if you want to progress, so let's say there is a clinical suspicion of FSHD, and you want to progress, you want to proceed with just, okay, I want to do it in a stepwise fashion. I want to rule out FSHD1 first. So then whole genome optical mapping can be ordered. And if that is negative, then the combined panel can be ordered, or the sequencing panel can be ordered to look for sequencing variants. However, if you want, it's your choice. And if you think that, no, let's go ahead with the combination testing right from the beginning, then that is also an option. So either it can be done in a stepwise fashion, which is what I meant as tiered approach.
Great, thank you. We have some questions coming in from the audience. So this one, maybe again, because the genetics is so complicated, if you can, there's a question about what exactly is the difference between FSHD 1 and 2? And so, I'm not, Yeah, go ahead.
Yeah, I mean, so it's a little complicated, right? So FSHD1 specifically is, as I said, autosomal dominant inheritance. It results from the repeat contraction of the D4Z4 repeat arrays. And if it's in that range, then that is FSHD1. FSHD2 is digenic, as I mentioned. So the SMCHD1 gene on chromosome 18, sequencing variants along with the presence of the permissive allele, which is the 4QA, not in the repeat range, in just the initial normal range, can also, and hypomethylation of that can cause this phenotype. So it's a little complex that FSHD1 is autosomal dominant and FSHD2 is actually digenic in inheritance. We've got two different chromosomes involved in leading to the phenotypic manifestation.
So which means that both of those would have to be inherited. So it's not just a simple 50-50 chance when you have just one gene involved. So,
right.
All right. And I think possibly people might be wondering also from a clinical, phenotype perspective, are they the same or different, FSHD1 and FSHD2?
So that's where if you look at OMIM, so there's a lot of phenotypic overlap, right? So it still comes under facioscapulohumeral muscular dystrophy. So you saw the common features that is where there is a little bit of ambiguity in a clinical diagnosis. So as you saw, the 14 samples that came to us, there was a clinical suspicion that some of these features or multiple features match FSHD. Now, is it FSHD1 or FSHD2 is what we were able to, essentially discern right now, which were FSHD1, and the two that were negative for this, they could be FSHD2. We still don't have the answer to that. We are trying to do some studies, but with all this COVID situation, that's been put on hold right now.
Right.
There are a lot of common features, as you saw, leg weakness and arm weakness and abdominal weakness, and then the scapular winging obviously is the hallmark that actually puts it into these buckets. But the patients may have some milder features, and then there's a little bit more complexity that I did not go into details is when you have, in the borderline ranges, then how can those manifest? The is that I did not go into details is that some of the newly diagnosed patients could be mosaic. So they could have the repeat contraction in some cells and some cells they could not. So again, the phenotypic variability, as I mentioned in the beginning, it's variable. We see a lot of phenotypes. All of them put together is where you want to have a diagnosis, a genetic diagnosis. Is it FSHD1? Is it FSHD2? Or is it a neuromuscular disorder panel? And that's where genetic testing plays a very important role, accurate genetic diagnosis.
Right. And can you just quickly mention in this panel, because people might not know what the concept of a panel is, but it allows you to test kind of in one go for many, many other genetic mutations associated with neuromuscular diseases. How many other conditions does that panel test for?
So for example, let me give you an example. So PerkinElmer Genomics has this neuromuscular disorder panel, which is an expanded panel, but let me give you a shorter one. So we have a panel which is called a limb girdle muscular dystrophy panel. Now that panel has 99 genes on it. And it is a sequencing panel where we're looking for sequencing variants or mutations in the layman word that could be pathogenic or that could be leading to the muscular dystrophy. Now, within these are also the DMD gene, the Duchenne muscular dystrophy gene, the EMD gene. So anything related to muscular dystrophy in literature or associated with the phenotype, because we've seen a lot of variability, become a part of this combined or neuromuscular disorder panel. So it's a lot of genes in there. And that is why it's called a panel, because a panel means that it's a range of a number of genes that are put together and we are testing all of them at once, if that answers your question.
Thank you so much. And not too long ago, people had to have a test done one at a time at considerable expense. So being able to test for 99 mutations or whatever in one go is
thousands of mutations from 99 genes.
Yep.
So yes, and then the cost of sequencing has gone down. So June, you're absolutely right. Traditionally, almost 2 decades ago, people were doing one gene at a time. It was very labor intensive, time consuming, and very, very expensive, one at a time. And now we have the ability to multiplex them or put all of them together in one pool. And now we've got bioinformatic tools that can give us the answer from, if there is a mutation from any of these 99 genes that could be pathogenic, that can explain the patient's phenotype. So now it's cost efficient, it's faster, and it's not that labor intensive either because of automation and the improvement in these technologies.
Incredible. Very nice. So we have a question from an individual who says the disease, the FSHD, manifested in me when I was in my 60s. Will it be the same for my children? They have never been tested and have not been told that I have FSHD.
So again, knowing the chance, right? So every one of your children has a 50% chance. Now, if you are a mosaic, then does that mean that, first of all, in your muscle and in your blood and in your germ cells, were you also mosaic? So were you able to transfer that in the mosaic form? And because you had late onset, there's no guarantee that your children will also have late onset. The manifestation can be anytime after adolescence. So that is where testing plays a very important role. And again, counseling, and again, how much information that's on you. You have autonomy for your genetic information and how you want to share that information with your family. I think I would refer you to discuss with your geneticist and genetic counselor and how to recommend additional family members get tested.
Because also it's possible in that situation that adult children may be genetically FSHD, and if they do not know if they're not aware of it, they can pass it on to their children.
Absolutely.
And we're aware of cases where the adult has a very mild manifestation, but sometimes the child even can manifest symptoms because there are many genetic other factors that can influence how severe the
exactly
process is.
So the variability is where everybody tries to be a little bit more cautious, that you don't want to take that risk because there is a chance. Number one, if it's autosomal dominant, you already saw in the initial slides, there's a 50% chance. So that's where it's very important to meet your geneticist and discuss with genetic counselor. They can put all this in writing for you, give you all the options, and give you every information that you need if you want to share that with your family members so that they can get tested.
Okay. We have a question asking if this test can be done in India.
This test is done in India. So PerkinElmer Genomics is a global lab. We have our lab in India, in Malaysia, in Sollentuna, Sweden, and our headquarters are in Pittsburgh. So we are getting samples right now. The Sapphire system is placed in Pittsburgh. So we are getting samples globally to test for FSHD.
Okay, great. So if you went to your doctor in India, they should be able to requisition it?
Yes.
Okay. Great.
Yes, they should be able to requisition it. And if you need more information, let us know. You can go to our website. We have a test requisition form, especially for FSHD. And that is the form that needs to be filled out. And we have some unique methods by which the blood needs to be shipped to us. So we will provide all that information, but we need to know ahead of time.
Okay. Several people have asked, how does the cost of this test compare with the traditional Southern blot?
I think I'm going to defer that question right now, but I think if you see on our website, the cost of the test is mentioned on the website. And I don't want to give a number that I am not clearly clear about what that price is right now, but you can compare that to any other traditional test. And I can tell you, I think it's almost 1/3 or 1/4 the cost. So it's a very cost-effective test that we have developed and established. And remember, because we have these populations that we want to help and get tested, like patients in India, patients in Malaysia, where we know that cost is a huge issue, this is an extremely cost-effective test, even for that geography and population. But it's available on our website.
Okay. Several people have asked if you were tested, let's say more than 10 years ago, or sometimes as long as 20 years ago, and at that time—
Please get retested. I'll answer right now.
Okay.
Please get retested. We've got newer, more sensitive, more efficient technologies. And because you have this available, you should get tested.
Okay. There's a question if insurance will cover the cost of testing.
We have, we are just looking into insurance billing right now. I know that there is a code for FSHD. Now, we are also looking into trying to get unique codes using this new technology. So I actually cannot answer that question right now, but to my knowledge that CPT code for FSHD that exists may or may not be used for insurance purposes right now. I'm not clear on that right now.
Right. I'll also mention that, you know, with insurance, often they will reject a claim initially. And if you come up with a strong case for their accepting it, they can, you know, we've been advised like you might go back 2 or 3 times to an insurance company to contest their rejection of a claim. And we have letters on our website to help you make that case, the scientific and medical rationale for getting tested, whether you have symptoms or don't have symptoms, but have first-degree relatives, et cetera. There's sound, in our opinion, sound medical reasoning for getting tested in those situations. So we have letters that help you make that case if your insurance company was to turn down the claim initially.
Geneticist, actually, that is what we are also encouraging, that the geneticist up front can also try to get pre-authorization from your insurance company.
Right.
Of medical necessity, as June, you were mentioning, that also will, you know, can play a very important role. So you will know upfront, and even later on, you can actually contest, and it is medically relevant. There's absolutely no doubt about that, and it should be reimbursed then.
Mm-hmm. Okay. So we have Amy Becker pointing out that clinical trials may, in some cases, require diagnostic confirmation in order for you to participate in the clinical trial.
Yes.
So that's another good reason. If you would like to be eligible to participate in future clinical trials, that is a good reason to try to get tested now. Let's see, I have a question. I live in Minnesota and my mom, sister, and niece have FSHD. How shall I get testing done? I have volunteered for research studies in the past, have never been called to participate. So I think the question there is what she's wondering if she might be a non-manifesting case, but she wanted to understand what her genetic status is.
So the straightforward thing to do is go to your geneticist, go to your physician, get yourself referred to a geneticist, give this information. If there is information available that your family members can share where they tested, do they just have the phenotype or do they also have a genetic diagnosis? Those 2 pieces of information are very important. And then your geneticist and your genetic counselor can then help you get the testing you want, just to absolutely be sure in terms of your, you know, offsprings or your family planning or what your genetic status is. That is the right way to go.
Right. Okay. So there is, here's a question. There's a test out there that's based on saliva I think they're referring to the methylation assay in Peter Jones's lab. Can you speak to the accuracy of collecting DNA information through saliva versus blood as your test requires?
So the straightforward answer is we have not validated that. So what we have done right now is what you see is what our claims are, what our performance is. And that is that we have validated this on a peripheral blood specimen. Now, what happens in muscle tissue, what happens in saliva, that is something that we would have to validate, get the samples from patients affected, and that would be the next round. So I cannot comment on what another lab has validated. I can only comment on what we have validated as the diagnostic clinical test.
Okay. There are a couple of questions here that are kind of related issues. So they have, questioner has a family member or, you know, an individual who does not have symptoms currently, but the parent has symptoms. You have an adult child who doesn't present symptoms, but is thinking about the future, probably especially about family, planning the risk of passing it along. How can such an individual get the test? Is that available to that non-symptomatic individual?
As long as you are an adult, so, you know, testing over 18 years of age, then the process, again, is exactly the same, right? So for any genetic testing, you need to go to your physician, get yourself referred to a geneticist, talk about the either clinical features or family history, and that you would like to get yourself tested, and they are the best people to counsel and order the testing. Now, if there is a minor, then the minor cannot do that. Again, parental consent is going to play a very important role. You know, if you are under 18 years of age, then again, the parents will need to go along with the child to the geneticist and then discuss everything and then see if they really need genetic testing.
Mm-hmm. Okay. So here's a question. What is the chance that an unaffected sibling of an FSHD patient could pass it on to their child? So unaffected, I mean, probably means unsymptomatic.
Unsymptomatic. Yeah. So until you do genetic testing, you don't know, right? So are you in the range of, so let me explain. So 1 to 10 is the repeat contraction. 7 to 10 is a gray zone area. 7 to 10, methylation testing or methylation is going to determine whether you're going to have FSHD1 or not. So if you are in the 7 to 10 range, you could be asymptomatic. However, you could pass it on to your child who would be symptomatic. So I think the only and only way to resolve this is to get genetic testing so that you know exactly which bucket you are in. Are you in the 1 to 10 range? Are you in the normal range? If normal range, then you don't have anything to worry about. Then not only are you asymptomatic, but you're also negative for the repeat contraction. So that's where I think there'll be a little bit more confidence as soon as you get genetic testing to resolve this.
Great, thank you. Let's see.
June, I just want to add that in the chat, I've posted a link to both the PerkinElmer website, which contains all information about you know, how to order the test, the cost of the test, all of that. So look in the chat for that link. And also I've posted a link to download a sample letter for the insurance companies as well. So just check the chat for that, grab the links and stick them somewhere, copy and paste them somewhere so that you have them handy for later.
Thank you. Thank you so much, Beth.
Sure.
Okay, great. Oh, there's a follow-up question regarding whether, you know, is there a rationale for testing if you're asymptomatic, I guess.
You just answered that.
Or mildly, yes. Or
Yes.
let's say if you're mildly symptomatic, you're not planning to have children. I think we addressed that before. If you are symptomatic, you know you might have FSHD. I think the question there might be then if you have any interest in participating in research or clinical trials, you would likely need to genetically verify that
Absolutely.
you actually have FSHD.
Yes.
So, and you have to weigh that against any possible negatives, like if you feel that might be lead to insurance discrimination or something like that. One would have to weigh that, I guess. But all of these things, as you keep pointing out,
Everything
Dr.
associated with patient autonomy and patients needing genetic testing, I don't think that discrimination holds true that much as it used to maybe a decade ago.
Okay. Let's see. Our people are wondering whether this whole genome optical analysis reveals any clues as to what accounts for the variation in symptom severity between individuals who have the same number of D4Z4 deletions.
So exactly. So, what we will see is the range between, let's say, 1 to 7, right? Or 1 to 6.
Yeah.
So depending upon what your repeats are, then that genotype phenotype correlation, which has already been well established, is there. But the variability could be because of n number of factors. So right now, the way that we are looking into this is that our analysis is very focused, which I did not go into many details about. So we are interrogating chromosomes 4, 10, and 18 by this optical mapping technology. However, if there are negative and if we intend to do any further research exercise, so remember, we validated this as a clinical test, but we have the data from the whole genome labels available. So later on, with consent from the patients and get this through an appropriate ethical review board clearance, then we can interrogate the entire genome to see if the number of D4Z4 repeats and anything else going on in the genome, or they themselves really can shed more light onto the variability in the phenotype and the severity of the disorder.
Okay. This is a follow-up. Before there was a question whether if you had previously had genetic testing, say with Southern blot technology, they wanted more additional information about what additional data would this newer technology provide?
So you have to understand that it depends on how the Southern blotting was done and at what time frame. Southern blotting is more of like, a manual estimation and what kind of methods did you use. So it has been considered the gold standard. So when we validated our test, just because there was no other technology available, we had to compare our data with the available Southern blotting data. But what we have seen from the experts, the European team that I showed you, those publications from Lemaire's group in Europe, is that they have seen a wide variability, in counting from Southern blotting methods. So what we have right now here, number one, with this optical mapping technology is one that it's a very novel technology. These digital images that we are getting and the mapping of these labels is highly accurate. It's completely consistent across labs and across runs. And so we have a lot of confidence that now we have a technology which is more consistent, more reliable, and we may change the paradigm of how to look at the data from, these other conventional technologies. So I do not have the answer to that, but it would be worthwhile to look at the report and see what methods were used and whether it is prudent to go ahead and do this new whole genome optical mapping technology on that same individual.
Great. Okay, thank you.
Discussion based on to look at the history of what methods were used. Is it really needed or not?
Okay. We had a question, if you could go back and clarify a little more what the terms mosaic and methylation and what the relevance they are. what their relevance is to the diagnosis of FSHD.
Okay, let me come back to the slide over here. So what is happening is that, you know, every chromosome or some of the genes, you know, there is a methylation pattern, or it's a phenomenon, like, you know, like we are talking about, how do I explain this? So methyl group on these bases that I showed you, right? Let me come up here, back here. Yep. So we can have the methylation or a methyl group attached to these bases. And this is a very unique pattern that happens in the human genome. And there are some methylation specific disorders that actually result because of this phenomenon, depending on where they are originating. Now, what happens is that methylation can either progress something or it can also block something. So when we are talking about normal gene expression, to say, like, you know, a normal protein to be formed, any modification to it, like methylation, can actually make it dysfunctional. This is the simplest way that I can explain. So now when we come up here, then what has been established is that if you have a permissive allele, which is here a 4QA, and if you have hypomethylation also of your repeat array, then that could then lead to FSHD phenotype. We are not doing that right now, as I mentioned to you, but that is something that we are definitely investigating and looking at. So this may shed another layer of light onto what is going on. The other thing that I told you is that if you have repeats in the gray zone, at that time also, if there is a specific hypomethylation, then you may have the phenotype, but if you don't have it, if the methylation is not there, then you may not have the phenotype. So this is the complexity and biology which is going on in the human genome. FSHD is just one of the disorders. This happens in cancers also. So when there is complete progression or, you know, an abnormal protein being formed, then this is a very unique phenomenon. So that's the methylation part. Now let's talk about the mosaic part that you said. So what is mosaic? Mosaic is a pattern, right? So you see different patterns. If you just think about the term mosaic, you have a little, I don't know, you know, if you go outside and you are walking on a sidewalk, you may see different sizes and patterns. So that's essentially the simplest way I can explain mosaic. So what will a mosaic do in FSHD? So if you have some cells that have the repeat contraction and some cells that do not, right? This is a very unique phenomenon that we have seen in a lot of genetic disorders. Again, inherited disorders and also cancer, because some cells will, you know, have the mutation and some cells will not. So cumulatively, that is called mosaic because you have a different pattern. Now, what could happen is that if you have a mosaic pattern, that means some of your cells have the normal, which we've actually seen in 3 cases. Let me show you the data. So what will happen is that if you have a non-mosaic, you know, normal individual, then you will have just this pattern. If you have a non-mosaic FSHD phenotype, then you will just have this contraction pattern. However, you may see both of these, and then we need to determine what percentage of normal cells do you have and what percentage of abnormal. And this is what I'm referring to as mosaic. And the mosaic will actually lead to considerable phenotypic variability, which you see over here. Some of the common symptoms and some of the milder symptoms and some of the unique, you know, just overlapping symptoms with other muscular dystrophy.
I might want to also ask you to clarify about, there's also something called a germline mosaic where,
so as I was mentioning that, let's see if I can, this one. So when we are talking about mosaic, so mosaic can be somatic. So somatic means non-germline. right? So let's say something just happened in your muscle, right? So you've got some cells that are causing the muscle phenotype and some that are not, and that is a somatic mosaic. But when we are talking about inheritance, we're talking about gonads of the father and the mother. So there could be a germline mosaic pattern that is coming from the parents, and that would then also could lead to mosaic in the offspring. So we're talking specifically about the germ cells here, the gonads, right? The sperms and the eggs, and which chromosomes in there are going to the offspring. So those are the germline mosaics.
Right. Great, thank you so much. Well, we are a little bit past our hour, so there's still many questions coming in, but I think we covered quite a few. So we want to thank you so much, Dr. Chaubey, and I will turn over the mic to Beth Johnston to close.
Great. Dr. Chaubey, that was awesome. I've been listening to this stuff about genetics and getting explanations for years and years and years and years. And that's probably one of the best, most clear, simple explanations I've ever heard. So thank you.
Thank you.
I will also note that Rena Vanzo, I think she might be with PerkinElmer, she posted in the chat a link where you can go to find genetic counselors to discuss the details of your testing. So please grab that link from Rena. Thank you for posting that. That's so that you can quickly find a genetic counselor in your area. Okay. So I guess with that, I'm just going to thank you for attending our Sequester Camp webinar today. And of course, a very special thank you to PerkinElmer and Dr. Chaubey of this excellent information and taking your time to be with us today. Just thank you guys so much. We appreciate you very much.
Thank you so much, everyone. And if there are more questions, Beth and June, please send them to us and we'd be very happy to answer them.
Oh, we will. Trust me. We'll be updating our Sequester Camp webpage with this video recording once we get it approved, and with all the other information as it arises. So please check back to our Sequester Camp webpage for more information. I've been posting it in the chat, so it's there. Next Thursday's webinar, we have another very special guest, Dr. Konstantin Pharmakidis. I know I totally wrecked that name. From the University of Kansas Medical Center. He's going to be speaking to us about adapting your home for safety. Very, very helpful. Bill and Mitch will be coming back this Monday afternoon at 3 o'clock Eastern, broadcasting their fitness workout. And they're going to continue their workouts every Monday through June 22nd, at least at this point. Also on Wednesdays at 2 o'clock Eastern, Belinda Miller's still broadcasting her weekly children's book reading on Facebook Live. And on Wednesday evenings at 9 o'clock, you can catch our radio show celebrity, Tim Hollenbach. He's on Facebook Live. This coming Wednesday next week, Tim's guests are actually Bill and Mitch. So be sure to turn in on Facebook or dial in to learn more and ask them questions. They have such a great program. Keep an eye out on your email for updates announcing all of our upcoming events. And we hope to see you again here next week. Take care, everyone, and please stay healthy. Thank you. Thank you again, Dr. Chaubey.
Thank you. Bye-bye.
Okay, bye-bye.